Hi everyone, I'm Shreya, a bioinformatics and AI engineer at Boltzmann Labs. Today I'm pleased to present our research on prioritizing biomarkers and genes in LUAD using graph convolutional networks. Cancer is the leading cause of death worldwide, so it's important for us to identify what change in the body caused it. But this is also quite a challenging problem since cancer is heterogeneous. It has many etiologies for a single phenotype. It's heterogeneous amongst patients and within a tumor, which is why it's a disease that is better characterized by multi-omics data. Multi-omics data can convey information not only about the features in a biological layer, but also about how these features interact within the layer and between other layers. To this end, we use graph convolutional networks, as they are able to aggregate both network information and feature information, thus providing us with a very holistic view of cancer. Specifically, we focus on epigenetic modifications as they are reversible, so they offer some exciting prospects for cancer therapeutics. We focus on lung adenocarcinoma in this study, and the multi-omics data sets that we use are gene expression, SNV, CNA, DNA methylation, miRNA expression, and protein interaction data, all into a single predictive model that is the GCM. From these multi-omics data, these are the important biomarkers that we identify. Most of our data is downloaded from TCGA GDC portal. We also download mutation CNA data from Broad Institute Firehose and gene expression data from the Genotype Tissue Expression Project. For the PPI network, we have used the string database. After we download our data from TCGA, we pre-process it. This will be discussed shortly. After pre-processing, we get a matrix that has 4 features per gene. In our graph, the genes form the nodes, and the interactions are derived from the PPI network. Our node classification problem, where we try to predict if each gene is cancerous or not, will be for semi-supervised training. This is because only a few of these genes or nodes in the graph have a positive or negative label assigned. This means that we know the gene contributes to LUAD, or we know for certain that the gene does not contribute to LUAD. The final graph that is predicted from GCN is a fully labeled graph. Each gene is predicted to be either a cancerous or a non-cancerous node. In pre-processing, we first concatenate all patient and case information into a single matrix for each omics data. We filter the samples such that we only have matched tumor normal samples. After which, we use 3 methods to perform imputation: mean, KNN, and DCA impute. We use the Python version of Combat called PyCombat to perform batch correction, and differential expression analysis is done using log2 fold change values. Finally, we compute the p-values to analyze the statistical significance of the nodes. As we can see here, methylation and miRNA data have CpG sites and miRNA probes rather than genes. So these have to be mapped to genes, and this is the methodology we follow to do that. Ideally, in our data set, we want the points to be clustered according to whether they are a tumor sample or a normal sample, and not according to the patient or the case file. Before batch correction, however, they are clustered differently. After batch correction, we see that the samples are being clustered according to whether they are tumor or normal. This graph shows us the cutoff for the threshold value and 0.05 as the p-value for statistical significance. So from this graph, we infer that 1.5 is the value for differential expression. Node masking. This is quite advantageous when we want to perform a semi-supervised node classification problem. In node masking, some nodes are masked, and when this node is a known node and has a target value, be it positive or negative, the model is able to learn on the problem better and accurately predict even the unknown nodes. The first graph shows us the intersection between the GCN predictions for positive genes and the target positive genes derived from this genet. We have taken the top 500 genes And here we can see that the GCN is able to recover almost 75 to 76% of the target genes when the prediction threshold is 0.75. The second graph is a heat map, wherein the first column represents the mean prediction from GCN trained on only gene expression data, and the subsequent columns are patient-wise gene expression. Using only the gene expression value from column 2, we get the top differentially expressed genes as 18. Using only mean prediction values from GCN, we get the top differentially expressed genes as 73. This shows us that even training a GCN on single omics data is far superior to using the data just as it is. The first graph here shows us the percentage feature contribution of all of the omics to the final prediction from the GCN. We see that without using miRNA data, just using DNA methylation, our feature contribution for epigenomics is about 16–17%. But using miRNA, this increases to 30, which is a significant difference as mentioned because epigenetic modifications are reversible. This is our training curve, area under ROC curve. Continuing our research, these are a couple of points that we want to implement so that we can validate and compare our model further. As discussed, our multi-omics model recovers 75 to 76% of known genes, a 10% improvement over current methods. Using miRNA expression data along with DNA methylation increased the epigenetic contribution to the recovery to 30%, which is a significant difference as epigenetic modifications are reversible. That's all from my side. Thank you.